Okay, so good evening everybody. So I'm Nicolas Mathevon from the University of Saint-Étienne in France, and I'm going my talk will be about human baby cries. So in my lab we have started working on human baby cries around 10 years ago. We started this project on baby cries with one idea in mind. Let's study baby cries as ethologists and bioacousticians. And so this is the journey that I will tell you today. So during this journey on human baby cries, many people have worked together and are still working together. So I won't give you all their names, but I would emphasize the name of David Reby here and Hugues Patural here, with whom I have started actually the baby cry project. So in vertebrates, parental care is often accompanied by striking acoustic begging displays by the offspring, and these displays can be extravagant in terms of energy expenditure, the risk of attracting unwanted attention. But yet, these vocal behaviors are a keystone of newborn survival. So we have the begging calls of birds, the distress calls of young crocodiles, the begging calls of pinnipeds, pups, etc., etc. Similarly, crying is the primary signaling strategy available to the human baby for eliciting parental care. So human baby's cry is a vital signal that informs about the state of distress and that participates in parental and caregivers' attachment. Okay, so it's an important signal, and it has not really been studied through the eyes of ethology and bioacoustics. So crying is a complex. It consists of sequences of complex acoustic signals that are produced by the vocal folds, the sound source, and at the level of the sound source, the frequency of vocal fold vibration determines the fundamental frequency, F0, of the cry, so the pitch of the cry. And then the sound waves travel through the vocal tract, and during this traveling through the upper airways, they are filtered. And this filtering leads to the formation of broadband components that are called formants. So basically a cry is a harmonic series with formants, and there can also be, and it's very common, some non-linear phenomenon, so biphonation, subharmonics, deterministic chaos, vibrato-like frequency modulation, etc. So those all those non-linear phenomena that are responsible for the roughness of the cry. So when we started our research on human baby cries, we decided to work on the whole communication chain, from the emitter, so the cry production, the signal, of course, so which kind of information is encoded in the cry, and at the receiver's level also, to see, for instance, what are the phenomena that can modulate the perception of information embedded in the cries. As in most vocal signals, some of the information carried by the cries is dynamic and related to the current emotional and physiological states of the baby. And besides these dynamic acoustic cues, in the cries we find also indexical acoustic cues. These indexical acoustic cues, they are related to the age, the sex, the weight, etc. of the babies, and altogether they form what we can call an individual signature. You know, these individual vocal signatures, they are very common in the beginning calls of birds and mammals, and for sure you remember the studies on parent offspring recognition in penguins or in pinnipeds. They were on this individual vocal signature. So it has been our first step to to investigate the individual vocal signature that is embedded in the baby's cries. So we recorded a bunch of different babies and we analyzed their calls, and here you have in a nutshell the results that we obtained. So when you do a multivariate analysis on these calls, it's very easy to distinguish between the cries of the different babies. Here you have the cries of baby 1. And here is the cry of baby 3. Yeah, so clearly each baby has its own voice. So there is an individual signature in the cries of the human babies. The next question is: do receivers perceive the cries vocal signature? Do they perceive this indexical cues? So we decided to test the ability of parents to recognize their own baby from its cries. So we tested 27 fathers and 29 mothers. Each parent was tested with a playback trial, including 2 sessions of 15 cries. And in these 15 cries, there were 3 different cries from 5 babies, and including the baby of the parents, and also cries from 2 unknown boys and girls of similar age. And the cries were of course presented in randomized order, and the experimenter was blind to the conditions. The participants knew neither how many cries were from their own child, nor how many different babies they originated from. And this is the result. So the blue box plot here presents the rate of correct recognition, so to say. So you see that we are really very, very close to 100%. The mean rate was around 90%, and there was around 17% of false positive. So parents saying that, hey, this is my baby, and actually it was not their baby. So the rate of recognition of their baby from the cries is excellent actually, and it's excellent in both mothers and fathers. There is not a significant difference between both sexes. And what we did, because previous studies that had been done in the 70s, they report that mothers do better than fathers in that case. But in these previous studies, they did not control for the time spent with the babies, and we did. And we found that if a parent, a mother or a father, spent at least 4 hours per day with her or his baby, then the recognition rate is excellent. So actually our results suggest an important role of experience rather than a sex-specific innate predisposition in determining a parent's ability to recognize their own baby from their cries. So women do not appear to have evolved specialized skills here, and parents of both sexes may rather make use of shared auditory and cognitive abilities. Okay, so now what we wanted to explore was the learning process. So we knew from previous studies in different mammals and birds that vocal recognition between mother and offspring, for instance, can require a few days. For instance, the studies on the subantarctic fur seal by Isabelle Charrier show this very well. But there's a lot of studies that have investigated this question in animals. But this has never been done with the human baby. So we decided to do this. In the first experiment, the first study was we did it in the hospital context with mothers who have just given birth. So we recorded spontaneous baby cries from 24 different babies in the context of bathing, when the babies were taking their bath, and then we tested the mothers with these cries. And the test consisted in a series of 15 cries, including 3 cries from the mother's own baby and 3 cries from other babies, 2 boys and 2 girls, of similar age, of course. Okay, so the playback were conducted in the room of the mothers at different ages of the baby, different days after birth. So here we have the result. So the x-axis represents the baby's age at testing. So here, for instance, 24 hours after birth, 72 hours after birth, and here we have the recognition rate by the mother. So what we see is that even only 24 hours after birth, the mothers are already able to distinguish their baby from other baby, because the chance level is 20% in our test. But what we clearly see is that the rate of success increases very quickly along the days. Okay, and within a few days, the mothers become really good at identifying their own baby from their cries. So it requires the learning process requires only a few days. Okay. We found also that an effect of experience on this learning process. At day 3 after birth, actually, the mothers who already had 2 older children or more were better at identifying their own baby, compared with first or second time mothers. Right. Okay. So now we know that the mothers learn quite quickly to recognize their own baby from its cries. 2-3 days are sufficient. But 2 questions remain. First, what about the fathers? And the fathers are very difficult to test because they are never here in the hospital just after birth. I don't know what they are doing, but they are not here. So it was not possible to test the fathers. So that's the first question. And the second question is this learning process a maternal thing? Or is any adult listener able to learn as quickly as mothers do? So to investigate these questions, we set up experiments in the laboratory, in the lab, and with non-parents that we made them becoming parents, so to say, because we trained them with cries with a given baby. So each tested person had to listen to 3 cries from an assigned baby. The assigned baby was different for each different tested person. The training phase was quite short. It was only 3 cries, and each cry duration was around 7 seconds. So 7 seconds of cry. And then after the training phase, a few hours later, we tested this person with a series of 15 cries that includes 3 cries from their assigned baby. So we tested it in 2 sessions separated by a few hours. And we obtained this result. We found that actually there is quite a big effect of experience of caring for babies. We found no effect of the participant's age, no effect of sex. But their prior experience of caring for babies significantly affected their performance at discriminating between their assigned baby and other babies. So you can see here the participants who had previously cared for babies less than 1 year old were more successful than the participants who never cared to baby. Their rate of success in recognizing their assigned baby was good. Okay, that's the first result. The second step in this experiment was to do another experiment where we investigated the effect of exposure on non-parents' ability to recognize a newborn from its cries. So we took non-parents, both men and women. We made them becoming parents by a training phase. They had to listen to between 1 to 6 cries from an assigned baby. The assigned baby was different between the different tested persons, and the duration of each cry was around 7 seconds. And then we did a test phase. We did exactly 2 sessions. One was immediately after the training phase, and the other was a few hours later. And in these test phases, the participants had to listen to a series of 15 cries, including 3 from their assigned baby, and they had to say, hey, this is my baby, or this is not my baby. So what did we found? We found this. Look, on the x-axis you have the number of cries that the participant had the opportunity to listen during the training phase. So here only 1 cry, 7 seconds of cries. Here 6 renditions of cries. Okay, and here you have the recognition rate during the test. What we see is that even with only 1 cry, so the person had only listened to 1 cry during the training session, the rate of recognition is better than chance, which is around 20%. This is the false positive. And after, if the person had been trained with 8 renditions of cries from the same baby, then it will be, he or she will be able to recognize this baby against other babies at a very good with a very good success rate. So this is very interesting. This is very interesting, and let's summarize the results so far. It means that yes, mothers learn to recognize their baby's cries very quickly, but non-parents achieve similar levels of performance in an experimental setting on the basis of very, very limited exposure. So overall, we show that neither the sex nor the parenthood status of participants impacts their performance at recognizing a baby's voice. Instead, our study emphasizes the key influence of the time spent with the baby, and also the influence of the prior experience in infant caring for all participants. Okay. So together, these observations suggest that baby recognition relies on both perceptual and cognitive abilities that are shared by all human adults, rather than specific to motherhood. Okay, so yes, there is some indexical acoustic cues responsible for an individual signature in the human baby cries, and listeners, parents, other caregivers can learn this individual signature, and that way probably they become familiar to the way each baby expresses its own need. But now, what about the dynamic acoustic cues? So the dynamic acoustic cues are very important. It's the second category of information that is coded in baby cries, and they are very important because they may code the level of distress experienced by the baby's instant needs, and possibly the cause of the cries. Okay, so to investigate the dynamic acoustic cues, we first decided to investigate how baby cries could code for the level of pain. And we did this experiment where we recorded the cries of babies in different conditions. First conditions was what we call the pain condition. It was during a vaccination session, so at the doctor's office, and at about 2 months old, when they are about 2 months old, the babies receive 2 vaccines, Infanrix and Prevnar. And Infanrix is known by the doctors to induce a low level of pain, and Prevnar is known to induce higher level of pain. So what we decided, we decided to vaccine some of the babies with Infanrix first, and then Prevnar, and other babies with Prevnar first, and then Infanrix. Okay. And we also recorded exactly the same babies in another condition, which is a discomfort condition, so during their bath. So we had these babies recorded during those vaccination condition and bath condition. Then we did an analysis, an acoustic analysis of the cries. So here we have example of pain cries, and pain cries we distinguished 2 phases during the pain cries. The first phase was the acute phase, so it was the phase the cries emitted during the injection of the vaccine, and the second phase was the phase the cries emitted a few seconds after, and you can clearly hear the difference. This is the acute phase, so there's a lot of roughness, a lot of nonlinear phenomenon in this cries. And here, this is the second phase, so a few seconds later. Sorry. So the harmonic structure is really present. There's less nonlinear phenomenon, and also the discomfort cries. So the cries recorded during bath. Yeah, so clearly there are some differences between those cries. So we did the analysis. We measured a bunch of different features, pitch features, spectral and temporal features, and clearly it's pitch features and spectral temporal features are both important for defining the acoustic structure of the cry. And we represented this acoustic space actually of cries, which is actually an acoustic space of pain expressed by cries. So here you have the bath cries. So PC1, yes, it's roughness, so the nonlinear features, and PC2 represents mostly the pitch features of the cry. So here the bath cries are in this area of the acoustic space, so they are with a harmonic structure very well defined. And here we have the cries that are emitted during the Prevnar injection, when the Prevnar vaccine is injected first. Okay, so it's quite painful actually. A few seconds later, the cries of the baby move here in the acoustic space. So they go in the direction of the bath cries, but they stop here. And then there is, for this poor baby, there is the second injection, the infant injection, and the cries move again towards close to the Prevnar cries. And a few seconds later, they go back here. But they stay quite far from the bath cries. Now, what happens for the babies that received the Infanrix first? So you remember the Infanrix is known to induce low levels of pain. Clearly, it's the case. Look, the cries induced by the injection of Infanrix are here, not here. They are between the bath cries and the Prevnar cries. A few seconds later, they move here, so they are quite close to the bass. And then the baby receives the Prevnar injection, but instead of moving in that area, the cries stay in the middle of the acoustic space. Okay, so clearly it's better to inject the most painful vaccine after the less painful one. And a few seconds later, the cries move here, and they become very similar to the bass cries. Okay, so it's interesting because we see that the acoustic structure of cries gives a good idea of the level of distress, level of pain that is coded in the cry. Now, what we found also is that the cry roughness is very important. It's the best marker actually to distinguish the level of distress that is to characterize the level of distress that is encoded in the cry. Now the question is, do receivers perceive these cries dynamic cues? So we asked them, we did some playback experiment, and what we found that the receivers, so both women and men, parents, are able to spontaneously use acoustic features of cries to discriminate broad differences in distress level. But what we found that they are not so good in differencing between, they are not very good to distinguish between small differences of cries. Okay, now I told you that we are interested in the factors that can modulate our perception of the information that is encoded in the cries. So what we decided to investigate a little bit, it's something that is very well known. It's the sex stereotypes. Indeed, we know that at adult age, the voice between men and women are on average very different. The men have more lower voices than women. And what we decided to test the hypothesis that adult listeners may overgeneralize these strong sex dimorphisms of men and women voices to their perception and interpretation of babies' cries. So we combined acoustic investigations and psychoacoustics experiments to test this hypothesis. First, when we analyzed the cries, so we recorded spontaneous cries from boys and girls at 4 months age, approximately, when they were given their bath by their parents at home. And then we analyzed the acoustic structure of these cries, and what we found that the acoustic structure of human babies' cries does not convey reliable information on the sex, and specifically there is absolutely no relationship between the pitch of the baby's cries and the sex. Here you have each box plot represents one subject, one baby, and the red are the boys and the purple are the girls, and you clearly see that there is absolutely no relationship between the pitch and the sex. You have girls that have very high cries and very low pitch cries, and you have boys that have very low pitch cries and boys that have very high pitch cries. And this is the distribution of pitch between boys and girls, and it's absolutely similar. Okay, so then we did playback experiments. And what we did, we played back to adult subjects cries that we have modified in pitch. So we use the same cries, but we fixed the pitch at different levels. So this is a cry with a pitch at 310 Hz. Now the same cry, but the pitch fixed at 440 Hz. And the same, but with a higher pitch. Okay, so each adult listener has to listen to a lot of cries that have been modified like this, and each time he had to say, okay, to tell us if this is a girl or this is a boy. He had to take a decision, and these are the results. Actually, this is the x-axis is the mean pitch of the cries, and the y-axis is the decision, so the perceived sex by adult listeners. 1 it's girls and 0 it's boys. What is very, very clear is that all the low pitched cries have been classified by adult listeners as boys cries, and all high pitched cries have been classified as girls cries. Okay, so actually adults classify babies' cries according to what they know about adult voices. So there is really a bias here. Okay, we also investigated whether these individual differences in cry pitch could have an effect on adult listeners' gendered attribution. So the level of masculinity or level of femininity that a listener could, attribute to a given cry. And here things are also very clear. When you tell to a listener that you are currently listening to a boy's cries, but tell me if this boy is more masculine or less masculine, and all the boys that have low pitched cries, they are told to be more masculine. And it's the same, or the reverse, so to say, with the girls. So if you use exactly the same cries, you use exactly the same cries during the playback experiment, but then you tell the listeners that these cries are from girls, please tell me if they are more or less feminine. All the cries that are very high pitched, they will be classified as very, very feminine. Okay. So, yeah, it's interesting, but it means that the perception of static cues of baby cries can be modulated by sex stereotypes, and probably they are modulated by many other factors. Now let's have a look on the perception of dynamic cues. Now we asked the people to rate the perceived discomfort that is encoded in the cries. So the people that we had two groups of listeners. One, they had to listen exactly the same cries, the same recordings, but for one we tell them it's boys' cries. For the other listeners, it was the same cries were told them to be girls' cries. And what we found that actually the listeners, they pay a lot of attention to pitch. Okay, the low pitch cries, they were rated as expressing less discomfort than the high pitch cries. So they pay a lot of attention to pitch. And it's a pity, because actually the pitch is very, very variable from an individual to another one. But so the conclusion is that when you don't know a baby, it does not really mean something. Where if the pitch does not really mean something, because you will have baby that will always cry with a low pitch, and you will have baby that will always cry with a high pitch. But nevertheless, we attribute the discomfort, the perceived level of discomfort to the cry. The other interesting thing is that there is a significant interaction between the sex of the listener, the declared sex of the baby, and the pitch of the cry. So female participants, they gave similar discomfort ratings to putative boys and putative girls' cries. But the male participants, male listeners, rated putative boys' cries as expressing more discomfort than putative girls' cries. Okay, I don't know what this interaction does mean in the everyday life, but I think that we should pay attention to this possible effect of sex bias when judging babies' cries. Okay, we have just seen that listeners pay a lot of attention to cry pitch. Yet, as I told you, pitch is highly variable among babies, and the main acoustic features that code for the degree of distress are not pitch-related features. They are the non-linear features, the roughness of the cry. In another study, a side study, I would say, we investigated the effect of this pitch-based assessment of cries by asking people to rate the cries of human and non-human babies. We had in our laboratory we had a data bank of recordings of chimpanzee babies' calls or cries and bonobo babies' cries, and so it was very funny to do a playback experiment with all these cries. So just listen to them. So the human bath, the human baby's bath cry, you already know it. Now the vaccine cry. Cry from a baby chimpanzee. And cry from a baby bonobo. Yeah, the bonobo, they have a very, very high pitch voice. So the chimpanzee babies and the bonobo babies, they have been recorded in very different situations. Some of the situations were extremely stressful, and some of the situations were not very, 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 very stressful. So what are the results? The result, here you have the mean pitch of the cry, and here the perceived distress. So there is a clear relationship still between the perceived distress by human adult listeners and the mean pitch of the cry. The interesting thing is that the bonobos' cries are the red ones. They are all high pitched because the bonobos, their voice is high pitched in any circumstances. And you see that all these bonobo cries, they have been rated out as expressing a very, very high level of distress, which is not the case actually, because in this recording some of the babies were not in a very, very distressful situation. So I think that this result shows that it's very difficult actually to judge the level of distress of an animal if you don't have any experience with it. Okay. Now, more serious thing, so to say. How can we find some correlates between cries, dynamic cues, and brain activation in human listeners? So this is something that we have just started. So I won't present you a lot of results, but just the first results that we got last year. So it's well known that the brain areas that are activated by real pain can also be activated when you see somebody experiencing pain. It's a phenomenon called empathy, and this is the cerebral basis for empathy. So as baby cries express different levels of pain or distress, we can make the hypothesis that when we hear a crying baby, this will activate the empathic areas of our brain. So we decided to test this hypothesis with 4 categories of adults: men without child, women without child, fathers and mothers. All adults of the same age and 20 persons in each category. And we decided to to do fMRI, so functional brain imaging, to investigate this. What did we find? We found that in all these listeners, pitch activates only auditory areas. So here we have, the brain in different views, and this yellow zone here are the primary auditory cortex. So pain level is poorly coded by pitch, actually. This result is in accordance with the psychoacoustic experiments I told you before, actually. Pitch is an acoustic feature that codes badly for pain. It's quite difficult to distinguish the different pain levels using pitch. It's a little bit possible, but not very. It's not a very useful parameter. Conversely, roughness, so the non-linear phenomenon that are very common in the cries expressing high levels of pain, roughness activates empathic areas. So the tempoparietal junction, the anterior insula, etc. Okay. So pain level is mainly coded by roughness. So that's very interesting because it correlates with what we found using the acoustic analysis. Roughness, the nonlinear phenomenon are more important than pitch for coding for distress or pain. It correlates with also the psychoacoustic experiments, and so everything makes sense, so to say. Okay. Now let's have a look on the different categories of person we have tested. Did we find some differences between men and women without baby, and mothers and fathers that have currently a young baby? Actually, we found mostly that there was the same type of cerebral activation in all brains. So mothers, fathers, as well as men and women without a baby, have a brain who is fully sensitive to the cries of babies. I would say fully empathic to the cries of babies. So the general activation of the brain is the same in those four categories. When we go into the details, we found that cry-induced brain activation is really not significantly different between mother, men without child, and women without child. And this is quite surprising, only fathers show a slightly reduced cry-induced brain activation. No men without child, but only fathers. And okay, we don't have to exaggerate things, it's a slightly reduced cry-induced brain activation. The brain of fathers is really activated by the baby cries, but is slightly less activated than the brain of those three other categories of persons. When we asked to, we don't have any strong explanation for this. When we asked the tested people to rate the pain level expressed by the cries, fathers were also the category of people who underestimated a little bit the pain level compared to the three other categories, the mothers, the men, and the women without child. So maybe men who become fathers pay a little less attention to the cries of their babies, on average, of course, because for sure there are big individual differences, or maybe they become less sensitive for whatever reason. We don't know. However, let's not forget the main result. All adult categories are sensitive to baby cries, and there is no strong influence of parental status. You don't need to be a parent to understand what a baby cries. Conclusion: human babies' cries contain both indexical and dynamic cues. Indexical cues result in an individual signature. Pain level is mainly coded by cries roughness, which comes from the non-linear features of the cry. Second conclusion, decoding abilities of baby's cries depends mostly on experience. Men and women show equal abilities to recognize a baby from her or his cries, and there is no significant difference between parents and non-parents, fathers and mothers. Only fathers may have a less empathic brain, but this has been to check. Okay. My last conclusion would ask the question: does the ethological bioacoustical approach allow to look differently to baby cries? Baby cry is a communication signal, and up to now it has never been really studied as a communication signal. So what now it's important to do is to try to understand this baby cry as a communication signal, to understand the information coding and the decoding processes. Another thing is that we know from other studies, from anthropology and from the evolutionary biology of the human species, that the human species is a cooperative breeding species, where it's not only the parents that take care of the babies, but a bunch of many other persons, even persons that are not related to the baby. So we have to, when we study the baby cries, we have to encompass the variety of potential receivers, the adults, but also the children and any potential caregivers. So there's a lot of experiments that have been to be set up to take into account of all the potential receivers of the human baby's cries. Okay, so baby cries are good indicators of baby needs and distress, and it is extremely interesting to study them. To study them, we need to record crying babies. However, I have to say that as all of you, I hope, I prefer to see babies smiling and laughing than crying. So just for fun, look at this video I found on the internet, and you will see that sensitivity to pain is highly dependent on the social context. Look how this doctor proceeds when he vaccines a baby, and see the result. Yes.
Oh, that is a juxtaposition.
Epic background.
Epic background, warm coats.
Yeah. You know, the weather here is awful. Winter is really starting.
Sure, Nicholas, thank you so much. And there are a lot of questions. It has generated a lot of interest, I think, between those of us who are parents and those of us who were maybe thinking of becoming parents. So look, yeah, brilliant talk. And I'm gonna kick off with the most asked question, which was asked by two different people. And the question was, very interesting talk, was a pleasure to listen to you. Could you elaborate on the roughness parameter a little bit more? What acoustic factors are included in it? Hang on just 1 second, because there's an extra add-on to that, which was, Andrea asked the same question, but she also says, does it include breath holding parameters related to a blockage in the chest?
Yeah, that's a good question. That's true that roughness is something that includes a lot of, actually it includes all the nonlinear phenomenon. So that includes, you know, biphonation, it includes noise, it includes a lot of things. So for the moment, actually, we did not really go very deep in that. So we just so we are in the process of investigating which exact acoustic parameters it includes, and what are the importance, the relative importance of all these parameters. Regarding the breath hold thing, I'm not sure I catch very well what it does mean. I mean, it does mean when the baby stopped breathing, I guess.
I think that's what she means. Yeah. So maybe
yes.
they just stop and hold their breath. Maybe a bit of
Yeah. So that is the same thing. I know when they are doing this, obviously there is no more acoustic. There is no more sounds that is emitted. So we did not take into account this kind of stuff, but for sure it has. The thing that I should say that it's very complicated to analyze baby cries because they are very complex. They have a very complex temporal structure. So it's very difficult to decide how you analyze them, and there is not only a single way to do this. So for sure, we made some choices to make things feasible, but I'm absolutely sure that we have missed a lot of information and a lot of things. So we have currently many people, many postdocs digging on this question and you know to increase our knowledge about the different acoustic parameters that are involved, especially the parameters that are involved in coding distress.
Sure, gosh. I'm just thinking back to your talk on baby crocodiles as well. I'm like, you work on all the babies. I mean, that's I thought I was lucky working on whales and dolphins, but you work on baby things. That is awesome. A question from Barbara. She asks, did you control for baby cry similarity between brothers and sisters when you say that mothers become better to identify baby cries when they have more children?
No, we did not do that. But for sure, it would also be very interesting to compare between the cries of the babies from the same family, because I don't think that any people have done that before. So we don't. Probably there is a genetic something in the cries of the babies, but we don't have any clues of that. So we don't know if brothers and sisters are more similar than stranger babies, and so yeah. So this is something that we would love also to record babies from twins, and that could be also very interesting. Yeah, because in the way the babies produce their cries, for sure there is some anatomical and morphological constraints. Then the more the babies are similar in their morphology and anatomy, probably their cries should be similar. But there is also a learning process. It does mean that the babies cry in response, or the baby learn to know who will respond and how he will get or she will get a response from the parents. And we know from, for instance, the bird literature that the begging behavior of the birds, the small tiny birds, they can modify their begging behavior very quickly depending on the way the parents respond to them. If baby birds can do that, for sure human babies can do also. So probably,
I'm like, yeah. Oh. You're saying that basically how we respond affects how they cry. So
I cannot for sure they have a big brain. So why wouldn't they do that? So probably they adapt their way of crying depending on the response they get. And that's another thing that has never been really explored, and it's very interesting. Yeah.
Sure, that's interesting for lots of different reasons. And
don't worry too much.
I'm like now reflecting. Okay, yeah, and for sure, my children, when they shout "Mom," it sounds exactly the same. I don't know which one does it, but it happens a lot. Okay, I have another question. I had it, and now I've totally lost it. It was a good one though. That was it. Yes. Okay. So not sure. This is from Ma, and they asked, not sure if I missed that part, but were the 3 recordings of assigned babies used during the test phase different from those 3 used in the training phase?
Yes, they were distinct, of course.
Okay.
So yeah, they were from the same baby, but it was not the same sequence of cries.
You're getting grilled here, that's why.
No pseudo replication.
Oh, yeah. Okay, and this is from Simon, and it says fathers have less empathetic brain responses. Could it be due to habituation or an evolutionary response or coping mechanism because someone in the family has to be able to get on with the background jobs while the baby is being cared for? That's funny because it was exactly what I was going to ask.
Yeah. You know, we don't know. I can agree with this kind of explanation, the evolutionary explanation, but I think we really don't know. First, the first, of course, this result is very attractive. I can understand why, but the first result is that the brain of fathers, mothers, non-parents, they are all very, very responsive to the baby cries. And the difference we found between mothers and fathers is a small difference. Still, it does exist. So why does it exist? We don't know. We should maybe because it's very difficult to know if it's something that is linked to a cultural thing. Maybe these fathers they were working at the time their baby were crying, so they had to, you know, maybe they had to go to work very early or whatever. So we really don't know. We really don't know. But there is something there that yeah. But I would love to do again. It's a huge experiment. fMRI, it's very difficult to do, and we included 80 persons on this experiment. So it's difficult to do it again. But it would be good to do it with different, I would say, different types of fathers. Fathers that stay at home, fathers that have to really go to work, etc., etc. Because probably we will find, you know, subtleties there.
Yeah, and also now with lockdown and people are at home a lot more, so there might be differences there as well and how responsive people are. I'm going to ask you one last question. We are running a few minutes late, but we're loving to speak to you, so I just we're being a bit naughty with our timings today. Last question. Great talk. This is from Helen. Sorry. Did you have a look in different species than humans at their empathic brain area activity to the baby cries, and how did you measure the level of brain activity?
Okay, so we did not look at the brain activity of the different species, but one thing we did, what funny thing we did, and we are currently writing the paper, we have played back human baby cries to crocodiles, and also bonobo baby cries and chimpanzee baby cries, and of course crocodile baby cries, and we compare their answer, and it's very funny because crocodiles actually they respond very well to the cries of all these species, and they seem to be very sensitive to the roughness in the cries also. So that's interesting. Probably there is, you know, it's not very surprising. We have been noting for ages, but there is probably kind of common codes between all these distress calls, so to say.
Well, look, I don't know about anybody else in the room, but whoa, it went disappeared. We respond very, very strongly to the cries of those little crocodiles. Yeah. So I can see there's a two-way thing going here.
It's something that should be very ancestral. Yeah.
Nicolas, I could talk to you about this all day. We're going to have to wrap.
Yeah, and thank you for the nice introduction. Huh? I'm not sure I deserve it, so thank you very much.
No, it's true though. It makes all the difference. So yeah, really, really grateful. And we will hopefully see you here in a few years' time. I don't know. We'll see how things go.
I hope so. I hope so. We normally we have planned to go to South Africa within the next few months, but you know,
yeah,
we are just waiting.
Okay. Okay, thank you so much. Okay.
Yeah,
on to the next session. Woo!